I'm joined today by a distinguished panel and very happy to have several Syapse partners up on stage with me today. So just starting from the right side, Jim Ford, who is the director of the Stanford Cancer Institute's Cancer Genomics Clinic. We have Lincoln Nadauld, the director of Cancer Genomics for Intermountain Healthcare. We have Tom Browne, the executive director of Swedish Cancer Institute. And we have Howard Skip Burris, the chief medical officer and executive director at Sarah Cannon, which is the cancer care operation of HCA. Now, the interesting thing about this panel is that each of these individuals represent organizations who have implemented or are implementing precision oncology programs. So they're here today to talk about their experiences. So I have the envious and easy job of just teeing up the questions for them. So what I want to do is have each of the panelists start off by telling us how have you set up your clinical cancer genomics programs, and most importantly, why are you doing this? So let's start off with Jim.
I'm Jim Ford. So I'm a medical oncologist and a geneticist at Stanford and have been there for several decades. And so I have a couple of hats there. One, the main topic of this panel, which is directing our tumor profiling and cancer genomics program and setting that up within an academic center. So I mean, I think that's my role on this panel to discuss the unique challenges in that setting. But we may want to come back at some point too. 15 years ago, I started the Hereditary Cancer Genetics Clinic at Stanford too, which is now thriving, and of course that whole field has advanced. And there's a lot of changes being driven by genomics in there too, that will, I know later in the panel, folks will be talking about multi-gene panels in terms of germline testing. So, so I think the current drive for genomics is very parallel to what that was 15 and 20 years ago, as the discoveries and the cost and the ability to do single gene germline testing came on. How do you translate that to patient care and show the utility? So for us today, that's really the case for tumor profiling. So why are we setting that up within an academic center? Well, certainly for the same reason that everybody else on the panel will discuss, because we hope that's good for our patients, and as medical oncologists trying to find unique ways to treat and individualize therapies particularly for us, solid tumor and lymphoma patients, to try to improve their outcomes is key. But I would say the other unique aspects of doing this within academics is also, of course, research, and to try to learn and disseminate information on this and try to really prove improved outcomes in that setting, which is a challenge for all of us. And finally, really to drive innovation and research within the institution as a means to identify targets and pathways for investigator-initiated research, and I can later give some examples of how that's already led to things, for example, from the previous talk in triple-negative breast cancer, and in others where the profiling itself is revealing targets that then are leading to clinical trials that we're involved in. So I think I'll leave it at that point. Move on.
Dr. Nadauld.
Yeah, Lincoln Nadauld. I'm Director of Cancer Genomics at Intermountain Healthcare, and that's an integrated health delivery system headquartered in Salt Lake City. It's comprised of 22 hospitals and about 180 physician clinics. And I joined Intermountain about 2 years ago after harassing Jim for a long time at Stanford. And I joined Intermountain specifically to start this cancer genomics program and implement clinical cancer genomics. Initially, we piloted our clinical program there in one geographic region of Intermountain Healthcare. And initially, it was set up as a referral-based program where patients were referred by their treating oncologist to the personalized cancer medicine clinic. Those patients then had molecular testing performed via NGS-based panel testing, which we have built internally and have a CLIA certified test, as others do. And then their results were reviewed by a molecular tumor board, and ultimately targeted therapies are applied either via phase 2 clinical trials or in some cases off-label treatment. And so as that grew and we started to see some outcomes, we expanded that and have made this available throughout the system and beyond the system, and have offered the testing services, including molecular tumor board and drug procurement services. We've made that more broadly available to anyone that wants to order that. The reason that we started it really was to answer this question of, if we implement cancer genomics for patients, what will the outcomes be? Will the outcomes be any better than using empiric chemotherapies, as we've done for decades? And as we move towards shared accountability, particularly in integrated health delivery systems, this is a major question. And so as we've gone down this path with patients and implemented clinical cancer genomics, that's been the major focus, is to look at the outcomes. So we've been monitoring, of course, obvious things like their survival, but also adverse events like ER visits and hospitalizations. And then we've done a very careful costing analysis. And that's one of the strengths of being in an integrated system, is that you can leverage all of that data. You know, we own the ERs and hospitals around, so we know exactly what patients are doing when they're accessing healthcare, and we can watch those costs, and we're going to be publishing all of that and presenting it at ASCO this year. But as we've monitored those outcomes, the trends are very interesting, and we can talk more about that, but I'll pass it on for now.
Thomas Browne, good afternoon. I'm the Executive Director of the Swedish Cancer Institute. The Swedish Cancer Institute is a hybrid institute. It's a non-university research practice. We have a 5-hospital system. We see about 6,700 new cancer patients a year within our system. And through an affiliation agreement, we're part of the larger Providence Health Services system. I've spent most of my career in academics, but on arriving to Swedish 2 years ago, we began a strategic planning process, and the core of that was really to come up with our concept in this hybrid environment, this non-university research practice environment, our concept for personalized medicine. And we came up with a dual meaning, the double entendre. Swedish has a long history of whole patient care, holistic approach to the cancer patient and their family. And that's now expanding, of course, through population health to mean in a population setting. So addressing socioeconomic, psychological, environmental, integrative health issues on the one hand. The other meaning, of course, is using molecular phenotypic data, in our case from the patient's tumor, to inform their management. Now, we are firm believers in the concept of not allowing perfection to be the enemy of progress. So we started with a modest, but in some ways, rather different approach. We put together with internal expertise, partnering with our exclusive anatomic pathology partner, Cellnetics. We began by constructing a 68-item, what we consider a highly actionable gene alteration panel that was launched a year ago. And in September of this past year, we launched an IRB-approved protocol, basically a registration protocol, that has 3 main purposes. One is to inform the prioritization of standard therapy. For us, the second and very important purpose is to match patients with available clinical trials, either within our system or elsewhere, and then finally to serve as a data mining source. What's different about our approach is we're intending over 3 years to ramp this offering up to eligible patients at the beginning of their cancer experience, so that within roughly 2.5 years from now, we anticipate registering within our system about 4,500 patients a year. So I'll stop there and hand the microphone over to Skip.
Thank you, Tom. Skip Burris, I'm the Chief Medical Officer at the Sarah Cannon Research Institute. As Jonathan said, the Sarah Cannon is now the service line for HCA, Hospital Corporation of America, and so I'll come back to that in a second. But the genomics program really began with the Research Institute, which has been a private, very similar as Tom described in terms of a research-focused private enterprise, looking at a core group of strategic sites that encompassed several hundred medical oncologists and approximately 15 to 20,000 new patients a year that put about 1,000 patients a year on phase 1 clinical trials and a couple thousand on phase 2, 3. Over the last 6 or 7 years, the primary focus of drug development, which is our area of research expertise and interest, has been around patients being identified by molecular subtypes and looking at mutations and alterations and various other aspects. So as that became a necessity, the first step was that every patient that was referred into the investigational drug program was profiled. And that worked in the short term, but the need became clear that as drugs are being approved and these trials now move to the phase 2, 3, to get our practice partners and our affiliated oncologists to begin to profile patients earlier on in the disease paradigm and then to look at clinical trials sooner, believing that participation in a trial should be the first option, not the last in fighting your cancer. That's been a tough road to go. The community oncologists have been hesitant to profile. I think, and one of the reasons we'll talk about as Jonathan moderates, one of the reasons to go to Syapse now is getting docs to profile and incorporate the data in their practice, to order the test, to be able to handle the results. The education awareness around that is really quite substantial. Now, in the last few years, HCA is the wholly owned Sarah Cannon Research Institute is wholly owned by HCA, so now the service line, Sarah Cannon, takes on the responsibility of those 155 hospitals across a variety of markets, now talking north of 100,000 cancer patients. And really the focus there is to bring the genomics platform to a place where all the patients across the system will be profiled at the appropriate time, and that'll enter into the decision making about what treatment should go forward. So you can imagine all the upscaling that needs to take place with regard to handling that data and bringing it into the EHRs and the like as it rolls forward. We had started out with and continue to use a local pathology group with which we're affiliated, the PATH group, that's actually now a nationwide company. They have a smart genomics platform, but I will say that the vast majority of our oncologists have been profiling through Foundation Medicine, and those results have been brought in. Pharma Biotech has accepted the Foundation Medicine panel for eligibility on trial, so that's been a nice win-win there. But again, the biggest hurdle is still getting the oncologist to profile. The vast minority of patients actually are still being profiled in our system.
So I think one of the major questions that people typically have is what is really clinically actionable today versus what's an exercise in research and drug development? And how does that impact how you set up your program? When do you do profiling? What type of testing do you do? Is the testing internal versus outsourced? So how do you think about the clinical actionability, and how does that inform the setup of the service? Whoever wants to start can go ahead.
So within the context of our IRB-approved protocol, as I said, it's a registry protocol. And as the data is gathered, of course, it has to be collected, organized, analyzed. Our protocol actually calls for monitoring of decision timelines, which those of us who are engaged in clinical research and clinical practice understand how challenging that is. But the intent is, at least on an annual basis, to formally reflect on how the molecular phenotyping has been used to impact on decision making, or just as importantly, not only positively doing something or in an action-oriented way, but deciding not to do something, which again is a challenging thing to capture. The other concept is that we have, just as has been suggested through other presentations, we have created probably about 20 gene alterations that we've put in the category of actionable, if you will, highly actionable. We have another category, and those would primarily, of course, pertain to approved indications for a drug and a specific target, if you will. Then in the applicable range, we have off-label usage as well as clinical trial activity, which again, for us, those two, that category, the applicable category, is probably the most relevant. And lastly, I'd connect to all of this. How many clinicians are in the room or cancer practitioners of one stripe or another? Not too many. A few more now after reflecting on what you do. So the point is that no matter, we all work at very sophisticated institutions. We're familiar with the expertise at those institutions, but it's striking, the acculturation barrier to this implementation is really striking. And no matter how expert, how well trained, how knowledgeable the physician group or practice is. And that's something that's been very striking in our experience.
Just to pick up where Tom left off, we've had the same question and we've noticed the same thing, and something we've watched is how does genomic testing change physician behavior and management of the patient. And we've seen that in fact about 50% of the time management of the patient is changed almost immediately upon receipt of genomic testing results, and about another 20% of the time the management of that patient is changed when they fail whatever treatment they were on at the time that the results were received. So we have found that to be very interesting. We think it's suggestive that genomic testing is worthwhile and has significant clinical utility. If you have any.
Just to echo those last 2 points, and Tom said it well, the enculturation hurdle is huge, and getting the physicians to want to see all the data and think through it. Jonathan asked about the actionable items, which, very few patients actually have the driver mutation, or they've got the target where you've got the Gleevec and CML situation, or maybe the EGFR mutated non-small cell lung cancer patient. But as we see the profile of a patient come in and you even match them to a clinical trial, we were talking about the perfect patient eligible for a study. So it's very common nowadays to see a breast or ovarian cancer patient who is the perfect patient for 3 different clinical trials at the same time, because they've got a PIK3CA mutation, so they're great for that biotech or pharma company with a PIK3 mutated cancer. They've also got an FGFR mutation, so they're perfect for that pharma company that's got the FGFR inhibitor, and yet they're MET amplified, so they might be eligible for the trial that's looking at combining something with one of the MET inhibitors, and yet that patient isn't the perfect patient for those 3 trials. She's really got a very different cancer. And so as we begin to assimilate and accumulate all this data and look at what's going on with these patients, I think looking at, and what's entered in the case report forms for many of these studies is often just the mutation that required eligibility, is really selling short what we learn about that patient and why or why not that therapy might be working.
So just a couple comments at Stanford. So, unlike all these guys in networks and hierarchical organizations, I'm in an exercise in herding cats. So the challenges are somewhat different, right? I grew up with a large group of medical oncologists and other clinicians, all of whom are above average, and all of whom are experts in their domain. So people are exploring genomics in many, many different ways, whether that's sending tests out to outside sequencing companies, whether it's in various research protocols that are giving genomic information as part of or independent of a targeted drug trial, et cetera, et cetera. So a real important piece for us in setting up a program, and what's been very satisfying working with Jonathan and the Syapse groups was how do we incorporate all this genomic information that's coming from different places and different formats in one place, so we could use that to ask other questions and not have it all siloed. And that's been very helpful, and there's many challenges we can come back to in terms of electronic health record and different systems and all this, but having a central repository for that data, not only for people we're seeing in Genomics Tumor Board and following, but ultimately to go back and ask some of these questions and be able to use all that data. Of course, also part of our program are a very academic bioinformatics folks that want to explore that data in more depth than we're using it, and just in trying to find a targeted therapy, but really that whole genome data that's potentially in there for other things. And then finally, you brought up the word actionability and where we're doing things. So those of you involved in medical genetics, germline setting, know that word resonates hugely as well. Their actionability is about a variant in your germline that leads you to be susceptible to a particular trait or disease. In this case, of course, cancer susceptibility. And there's been an enormous shift in that in just the last year from single gene testing or just a couple like BRCA1 or 2 for clear syndromic families to doing panels of 20, 30, 40 genes that span all those panels and even include moderate dependent genes for which we don't fully even understand the risk. And multiple companies doing that. Of course, the whole scenario has changed with the Supreme Court ruling on patents. So things are shifting very fast there. Many opportunities for users in different places to do that, and interpretation of these variants in terms of actionability for risk. So there's a real paradigm shift there too.
So how do you think about the reaction of the payer community and the pharmaceutical community to the programs that you're developing? So are you able to procure drug? Are you able to get your testing reimbursed? Does this differ if you're talking germline versus somatic? So how does the payer side figure into that? And then especially for Skip, since you're a very, very large system, how do you think about the financial implications? And part of a public company, how do you think about the financial implications of these programs?
Well, in terms of the reaction from payers, we're seeing that payers are willing. Initially, we ran into problems with procuring drug occasionally, and their behavior has changed, and so that's interesting. I think as they get on board with understanding how this testing works, what the implications are, and what the clinical outcomes look like, we've frankly seen a change in our ability to procure drug for patients. So we have a much easier time now than we did a year and a half ago when we started, although it wasn't. I like to think that it's due in part to the some of the heavy work we've had to do in communicating with and educating the payers. They're also reimbursing for testing largely. So that's good. And then pharma, the relationships with pharma have been very warm. So there are many pharmaceutical protocols out there that dovetail perfectly with these clinical cancer genomics programs that you've been hearing about. And it's very easy to say, well, I've got this patient with this genomic profile with these alterations. They are now eligible, as Skip mentioned, for these 2 or 3 different protocols, and we can just pick the one that we want. And so the reaction from payers and pharma has really started to enable this, the implementation of cancer genomics.
Right, I would echo Lincoln's comments. I think the early reaction that many of us have had has been one of cautious surprise, but indeed the reimbursement patterns are all over the map, as has been described earlier today. I think the usual gray zone is that where what is medical necessity and what is in a clinical trial context, and in this new era of molecularly driven therapies in the investigational setting, where increasingly the eligibility criteria are defined by some degree of molecular profiling, how are we going to integrate the concept of supporting clinical research through third-party payment or not? And again, we've heard a little bit about that issue earlier today. So again, I feel very strongly that the issue of equipoise, we're beyond that at this point, because equipoise is a concept that really is driven by randomized clinical trials, and in most of these settings, that's not what we're talking about.
The payers have been, I think in general, very receptive. As Tom said, they've been a little all over the map, but there's very few outright no's. I think often it's been the insurance plan that the patient's chosen, which might derive how the benefit rolls. But that being said, we've actually talked to a number of the payers who've been supportive of some of the clinical trials we're trying to have roll forward. There's a couple out there now. We've got a trial called My Pathway that will be updated on at ASCO, where we have Genentech's allowed us to take their portfolio of approved drugs, and if you've got a patient that's eligible through one of those aberrations, you can enroll on that basket study and receive that therapy. When we're trying to do that as much as possible, simply because, as Lincoln was alluding to, if you don't do it that direction, you could have a lot of one-off single patient phase 2 trials. Unless that data gets pulled together, really hard to judge the benefit for the masses. The other part of it, I think, which is interesting, is the imaging of a patient from the CT scan, PET scan. So you go into staging a patient for their initial workup, and you're in for several $1,000s at a minimum, and then you think about the cost of these tests and pick your price point, but in the range of several $1,000s. If you're a payer, would you not mandate, before we started writing for $10,000 drugs, that you had that bit of information to think a better decision could be made? We're trying to get up the food chain, certainly having all relapse refractory patients profiled before taking that next step, and then certainly thinking in many of the solid tumors it makes sense to even get further up into first-line therapy and think about profiling at that point, putting all that data into the patient's paradigm for treatment and seeing how that rolls out. So very much a moving target, but I think people are receptive, and I think the more we can integrate the data, report on it, the more likely we are to be successful with the next steps.
So to extend one of the points that Skip was making, one of the concepts that many of us who have worked with fellows and residents, house staff over the years, one of the key concepts for a young physician is sometimes the most important thing that you do is what you don't do. And of course that in some ways is the key to, I think, this relatively early positive reaction by third-party payers, because indeed much of the decision making is in the realm of not doing something that ordinarily you might have done. So I think that's important. The second comment about moving molecular profiling to the beginning of the patient's experience, again, we've really bought into that concept in the sense of feeling that over time, not right now, but over time, that molecular profiling will inform the entire arc of managing the patient's experience. It's not the the ultimate answer, but it's one more tool to assist in the patient's long-term management, to include survivorship, et cetera.
Yeah, just to extend on that, we have seen a very similar trend. When we initiated our cancer genomics program, we found patients getting this profiling genomic testing done late in their course, and oftentimes, unfortunately, it would be frankly too late. The testing would be ordered by an ordering physician kind of at end stage, and the patient would subsequently decline, and we'd never be able to act on that information. As we have seen some of the positive outcomes, that has naturally moved up, so we're seeing ordering physicians now getting this testing early in a patient's course. And in fact, as we've monitored the outcomes across the entire system and with shared accountability requirements in place, we're now making it a system-wide goal that all patients with advanced cancer get molecular profiling at the time of diagnosis, because that provides a substantial roadmap for the overall patient treatment course. So there aren't any surprises as you go along, and that's really guided by genomic testing.
So to that point, how does shared accountability impact these programs? Is this something where these programs are now going to be a necessity for many hospitals? Is shared accountability going to bring about the age of genomic medicine? And if so, how do you think about tracking the outcomes, improving the clinical utility and cost effectiveness?
Well, I guess I'm holding the microphone. So,
the question of the day.
Yeah. It's all about outcomes. And so if patient outcomes are superior, then of course that's what we're obligated to do. Within an integrated system where we live, the key is we're seeing not only patients living longer, but also having fewer adverse events, and there are other positive trends. And so if you can decrease the number of ER visits, for example, and decrease the number of inpatient hospital admissions due to side effects from treatment, then it would make sense that your overall cost of care goes down. And so that means the entire system wins. And so that's where the motivation has been in our particular program.
One of the side projects, one of the fun things about initiating any program is devising relevant, tangential, but important projects. And one of ours entails a collaboration with HICOR, the Hutchinson Institute for Cancer Outcomes Research, Scott Ramsey and his colleagues, and we're attempting to evaluate the value proposition, just as Intermountain Health is doing, for this approach. And it has everything to do with the details that Lincoln has alluded to. And I think this is an area where there are going to be some surprises in terms of what we glean from outcomes research overriding the entire personalized medicine effort.
And so, Jonathan, going back to your accountability point, and Lee Hood was talking to me about this morning, the patient who comes in and they assume it's going to be a positive outcome, so the cancer is behind them, and then they want to get on with survivorship, and several speakers brought up the fact that there's almost 15 million cancer survivors in this country. So what we've been trying to do is, at the time we meet the patient, like Tom alluded to, having the patients consent to a registry protocol as appropriate, maybe profiled or not, but certainly get them engaged to share their outcomes from the very beginning. We time in with a nurse navigator. We've got a couple hundred nurse navigators. We'll have 600 hired over the next few years. So at the beginning of that patient journey, they're part of the process, and call it the satisfied customer or whatever that relationship is. I think that we're going to have that long-term relationship with these folks. People are living longer, and keeping track of their medical story and how they've fared in terms of their outcomes will be very key.
So, as you know, within academics, cost is very hard to evaluate because there's such heterogeneity amongst everything, but particularly amongst reimbursement and patients, where patients are coming from, and all these things. So I think we very much are concentrating on the utility side, right? Trying to, how best can we study this to show improved outcomes as part of this equation. And that's a real challenge to the traditional ways of studying outcomes, particularly in treating metastatic cancer patients with relatively rare cancers. Now we're becoming amazingly rare because, of course, you're starting to talk about genomic subgroups. That traditionally has been large phase 3 multi-institutional trials that could be spread across many sites in a very organized fashion. It's very hard to design anything that way here, of course, because you can't randomize in a dichotomous way, but also because all the genomics programs have been built within different institutions, and there's so many changes. So a real opportunity for academics is to bond together and share data in various ways. We've worked with Syapse about having a common platform that can be used in different settings, and not just academics, other folks in networks like this, where we can each bring to the table something. It might be simply volume and a huge access to large data. It could be particular research questions, very focused of looking at that data altogether in with various levels, of course, of HIPAA compliance and security for patient individuality. So I think that's a major issue, and lots of interesting ideas going on about how to do that. The other point is, of course, the NIH is sponsoring new trials that will hopefully address this in a very large aspect as well. One coming up that many sites, hundreds or if not thousands of sites will participate in is the NCI Match program. Again, basically a silo-type basket trial design with many drugs that pharma, particularly pre-approved drugs in the pipeline that pharma is contributing through CTEP in an intergroup type fashion across the country based on a genomic analysis that'll be done centrally through the NIH. And so the goal there is to put 3,000 patients on trials to look for outcomes based on genomics and powered to find some differences. So that's certainly happening in the academic setting.
So how do we scale all of this up? So we have lots of complex information being delivered to physicians. I think, Lincoln, you may have mentioned earlier that you have lots of community physicians that you need to deliver this complex decision support to. So how are we going to scale the system up Do you buy into the argument that sharing more data and aggregating more data on more patients improves this? And is the electronic medical record the answer?
Yeah, so I mentioned before that all of the molecular testing that's performed on patients is reviewed at a molecular tumor board to provide some guidance to our colleagues in the community about what treatment options that patient may face. And this is a multi-institutional molecular tumor board, and Jim and others from Stanford are on it. It's hosted and anchored by Intermountain Healthcare. Some of Tom's colleagues have been on the molecular tumor board, and it's really powerful to have brilliant minds and experts in genomics from multiple diverse institutions think about each patient's set of molecular findings and what treatments might be available to them. To scale this up, we have to band together. You know, we frequently find ourselves in this position of saying, well, here's a colon cancer patient with whichever molecular alteration, and it's the first time we've seen it, or maybe we've seen it one other time, and so the N is always 1. And to get away from that problem, we have to aggregate data so that we have big data to really determine what outcomes make sense. And so, you know, Stanford and Intermountain have been doing this, and we're building a consortium of institutions that will share de-identified data in an appropriate way, so that when I have that unusual sarcoma patient come in with an unusual molecular finding, I can search that database and determine whether Jim has seen something similar at Stanford or any of our other colleagues at these other institutions. And the key to all of that is having a common platform that you work on from an IT standpoint. And so actually John and our colleagues at Syapse are helping us do that, frankly. They're helping us build that common database. So that is very powerful, to aggregate big data together to make decisions about how to treat patients.
Clearly, the other aspect of that, the other commonality that really gives impetus to achieving that large critical mass of information is to have some consensus on what the highly actionable targets are, what the modestly actionable targets are, et cetera. Just as Dr. Klausner so nicely reviewed in terms of this recent consortium effort in defining different categories of actionability, if you will. The other aspect, and it's a little tangential to the subject, but it's one that's intriguing for most of us who've been practicing, especially in academic settings or tertiary quaternary settings for a while. We've all been trained and have done research in the disease site-oriented fashion, and many of us have anticipated the day when the site of origin of a tumor would be trumped by the molecular phenotype, if you will, of the patient's tumor. And are we there? Obviously not, but it's amazing how rapidly in some areas this is unfolding. And how will this impact the way especially specialty centers function individually and as groups? We're finding this with our molecular tumor boards, that you want to be sure that you're cross-cutting the disease sites. We have 9 multidisciplinary disease site teams that have regular conferences and their prospective case reviews. And we're ensuring that our separately organized molecular tumor board is engaged with those disease sites.
Yeah, I'm smiling thinking about a few years ago when we were pushing the profiling initially, and some of my colleagues were worried that, they're going to push a button, order a test, and be told what to do with the patient, that it would be cookbook medicine. And it's been actually the opposite. We're not getting them to push the button, but when they do get the answer back, they're, gee whiz, now what do I do with this patient, which is the molecular tumor board and the like. I mean, it's made it more complicated. Tumor heterogeneity, what biopsy to look at, rebiopsy the patient, how should that all be incorporated with what's going on? And as Tom and Lincoln both said, pulling together experts. I mean, the education, awareness, scaling this up, and Jonathan gone to Syapse. I mean, it's going to be key that you've got that systematic approach to get it ordered, have the data come back in, and make that less of a burden, so at least we're at the place where that's sitting in front of these physicians and they can reach out and have the conversation about what to do. All the profiling, all the things we've learned have made it much more difficult. It's not just the 200 different diseases you could subdivide cancer into. Now it's another factor on top of that when you layer in the molecular aberrations on top of all the histopathologic features.
Right. Just to emphasize again, said another way, the integration into the clinical workflow, into the substantial burden that most clinicians feel with whatever EMR or EHR they happen to be using is key. So to have an IT platform that clearly integrates into one's existing EMR is critical.
Yeah, and Tom, that reminds me, I mean, that's a great analogy because not many clinicians raise their hand, but as a medical oncologist, we all know when you get that 3-page radiology report or the 5-page pathology report, you go to the conclusion. And then maybe you'll flip back through it. And with this profiling information, very little conclusion at the end of it, right? So how you bring it into the workflow and make it so you can assimilate it, really, really going to be important for the clinicians.
So just a slightly different take on the scaling up question and idea. As intuitively attractive as this whole approach is in terms of targeted therapies and personalizing to a genomic signature, we've yet to even demonstrate the improved outcomes in that in a general way outside of some specific examples. And we all have anecdotes of really remarkable responses with single drugs to targets. It's clear that we have many more anecdotes of that not being particularly effective or not for long. So you can already see that this is going to quickly move beyond a single drug, single target paradigm, at least for common solid tumors, and get into the far more complicated area of putting multiple drugs together based on multiple genetic changes, which is what we're seeing in these tumors, of thinking about drug resistance, of how do you combine immunotherapies with these targeted agents for which we don't have great biomarkers yet. Lots and lots of clinical research questions. So I'm not out of a job yet. So as always with cancer research is how do you incorporate this most recent change into the larger future picture?
Right, and to extend on that point, I think most of us would agree that the modern practice of medical oncology has at its base clinical investigation. And to me, it is somewhat unlike many other medical disciplines that is outside of cancer. And I think that point is that that innate sort of cornerstone that is clinical investigation is only increasing in importance in medical oncology because of what we're describing.
So one final question for Skip. So you published probably about 6 months ago that you've sequenced about 2,000 patients and you reported out some early results. But Sarah Cannon as an organization treats about 100,000 new cancer cases a year. So how are you going to scale that up, and anything you want to announce about that?
So again, we've made the decision to partner with Syapse, so that'll be our choice software application to in fact put into our systems across the country. And Andy Kortz, our Chief Information Officer, and Holly Dilks, Dr. Dilks, our Head of Personalized Medicine, are here meeting with Syapse, and that contract's been signed. That'll be the implementation, and we'll move forward with, in fact, getting a program in place so the physicians will be able to do the ordering, have the results come back in, and have it assimilated and incorporated into their patient management from the very beginning, following clinical protocols and the programs that are set out. So very exciting next step for us, and again, trying to get those patients profiled closer to home and have that data analyzed by experts.
So 1/6 of the cancer or 6% of the cancer population in the U.S. will receive profiling soon.
Yes.
All right. Well, thank you everyone for coming, and I hope you enjoyed the talk.